all of this. So there's so much stuff that I have to say, okay, we're going to talk about this, but you'll learn more about it later. So please bear with us. I promise by the end of the program it all comes together, or at least it should. Okay. So differentiating leukemias, we don't just look at the morphology. We talked about how do you tell the difference between a lymphoblast, a monoblast, and a myeloblast. They're all blasts. Okay? Very, very few. I mean, sometimes you can get lucky. There's something called an hour rod, and we'll talk about those later. And if those are in there, those are kind of a little sign that says myeloblast, and you're like, yay, I can tell these apart. Other than that, pathologists who spend, 5 years in their residency can sometimes tell the difference, but a lot of times even they can't. It just is one of those things. We have to have some other ways to tell them apart. So that's what we're going to talk about today. This is the piece that really has come forward in about the last 10 to 15 years. Prior to that, it was all pretty much, we're going to stain it, see what it looks like. We used to use cytochemical stains, which we found out later were highly carcinogenic, terribly, induced birth defect. I mean, these were horrible, horrible things. And we used them all the time. But we found some other more sophisticated, more specific ways to differentiate these out. So we have to classify, and this is acute leukemias. It works also on chronics. Definitive identification, you need a bone marrow aspiration and biopsy, unless the patient has enough circulating blasts. We've had several patients, the pathologist has said, we really don't need to do a bone marrow since her peripheral blood is 90% blasts. I think we can probably figure out what's going on based on what's in the peripheral blood. So the woman had a phlebotomy, had just a simple venipuncture instead of having, a very large needle shoved into her bones. We look at the morphological identification. What's that?
What they look like?
What they look like. What shape they are, exactly. What do they look like? What shape are they? Immunologic and cytogenetic. This we're going to be looking at what markers are on the surface, what proteins are on the outside of these cells. And cytogenetics is just that. We're going to look at the genetic makeup of the cell. All right, we have to do a little bit of a serology primer here. Definitions that you have to know, and it's like the most circular definition on the face of the earth. An antibody is a protein that a B cell lymphocyte makes in response to a stimulus. And an antigen is the stimulus which sensitizes B cells to produce antibodies. Antibody generating. That's what this stands for. Okay? So basically your body is exposed to something, or these antibodies are exposed to something that is foreign, and they make an antibody against it. And we'll talk about how we're going to deal with that in a moment. An epitope, honestly, if you don't remember this word for now, we'll hit that in the fall and I'll worry about it then. That's the specific piece of an antigen. Okay. Oops. What is not an antigen? A liver is not an antigen. A protein on the liver may be an antigen. E. coli is not an antigen. The OX19 protein on the surface of the E. coli is an antigen. Okay, it's a small piece. Antigens tend to be a little bit, at least molecularly speaking, they're somewhat smaller. Okay, it's not a whole thing. It's just a protein or a lipoprotein or a glycoprotein that is going to stimulate your immune system to make antibodies. Okay? When you get the flu, you don't make antibodies to the flu virus, you make it to a capsid protein or a specific piece of that. That's the antigen. Okay? The antibody is what your B cells make in order to fight that off. Well, we kind of hijack that. And we use it in the laboratory to test with. Because if I know that this antibody reacts with this antigen, and I put them together and I get a reaction, guess what? I know both things are there. Okay. Cross-reactivity. Each antibody is specific for a single antigen piece, an epitope. However, you know when you're doing a puzzle, and you're putting the pieces together, and you know, that one, I think that one fits, and dang it, if I pound it hard enough, it'll go in there. Okay, even that's not really what's supposed to be there. If you really work at it, you can get it to work. That's what cross-reactivity is. I'm not going to spend a lot of time on this, except that you need to know this sometimes is cross-reactivity. If two antigens are very similar, I can get a false reaction. Okay. We're going to go into this a lot. The older antibodies we used for serology were polyclonal. They literally had farms of animals, goats, rabbits, horses, we used a lot of horses, that they would inject with just the antigen, and then they would draw blood from these animals, not necessarily kill them, because they could, if they were alive, we could drain them for a long time. They would draw blood from these animals, purify out the antibodies, and use it. They're produced by different clones of B cells, okay? B cells have the ability to clone themselves. It's the only mature cell, we talked about this when we did leukopoiesis. Lymphocytes can't make baby lymphocytes, but they can make clones, okay? And each clone is absolutely identical. In a living organism, when we're producing these polyclonal antibodies, you're going to get different B cells making these antibodies. So each one may be just a little bit different, okay? We're all human beings. We all have the same basic DNA, but no two of us are exactly alike, right? Even identical twins are not exactly alike. There's always some little nuance, some little thing going on. So they may react with different epitopes of the same antigen. That's not a good thing. We like to have nice, predictable stuff. So what we actually do, we now make something called a monoclonal antibody. We take a B cell and we say, make this antibody. And once it starts doing that, we cross it with a myeloma cell, which is a rapidly dividing cancer cell. What we wind up with is a hybridoma cell. Basically, we grow lymphocytes in a vat. We put them in a great big vat. We put all the nutrients they need in there, the proteins, the sugars, and everything else. And we have all these little clones swimming around in our little vat. Take a little antigen, we drop it in there, and it's like little piranhas. And they all start producing antibodies. They're identical. So the antibodies that they produce are identical. So we're going to have a much more steady reaction, a much more predictable reaction, and in most cases, a much stronger reaction. This is going to get really big in blood bank. Blood banking, we used to have a lot of cross-reactivity, a lot more problems. Once we went to monoclonal antibodies, boom, many of those problems went away, not all of them. Okay. So do you understand what we're doing here? We're making an antibody to something in particular, okay? If I want to make an anti-Cris antibody, I can take a piece of Cris, drop it in my little hybridoma cells, and it will make antibodies against Cris. Close enough. Do you understand? So I can say, I want you to make an antibody against this. And if I have sequestered these things so that they live in a vat, they're not exposed to anything else, right? In a horse or a goat or a rabbit or whatever else you're making them in, or even an old style cell culture, they might be making antibodies to something else as well. So a lot of people in the 60s and 70s especially, had anti-horse antibodies. Why? Because a lot of stuff was given in horse serum. So you do their testing and they react with everything because the antibodies we were using were created in horses. So you get cross-reactivity, you get impure reactions. When you go to these monoclonals, the only thing they've ever seemed to be exposed to is what you gave them. So they're very, very specific. This is basically what an antibody looks like. This is the IgG molecule. There's some others. Essentially, you have a constant end. This end doesn't change. It's nice and solid, stays that way. Then you've got the antigen binding sites up here. Those are sort of like Play-Doh or silly putty. When something gets on them, they mutate so that they wrap around it. They use the key in a lock so that it only fits one lock, or they are very specific to it. And so you've got your solid end, which does this, and you all know I can't draw or squat, so shush. Then you've got this end, and if I get my antigen that comes in here, okay, this end changes its shape to match. Okay. Then if I take away the antigen, and it's going to produce more than one of these antibodies, now I've got this little piece that exactly matches my antigen, and it goes out there, and anything that fits in there, it grabs onto and won't let go of. Now when it grabs onto something, I can't see that. It's invisible. However, I got this end down here that ain't doing nothing. So I can tag stuff onto it. I can attach an enzyme that's going to turn blue, or a fluorescent dye, which can give me a variety of colors. Or I can hook it to a latex particle. Remember the D-dimer test that you did? Okay. All that was, took a latex particle, coated it with antibodies against your D-dimer, and so it went through and it globbed onto any of the D-dimer that was in there. Okay. And when it did that, these particles stuck together and you could see it. That's how we do serology testing. And again, we will talk about that ad nauseum later. So we take these tagged antibodies. We actually can add multiple ones at a time if we add multiple tags to them. So like if I tag one red and one green and one blue, then I can look for red and green and blue, and I can test for multiple things simultaneously. I have to use some sort of visualization. Somebody with a lot more time, a lot more funding, and a lot more patience than I have, went through and figured out that each cell and each cell type has a variety of proteins on its surface, and they named them cluster designations, or CD markers. Okay? Each CD marker is specific for a particular protein. I'm going to put a few of them up on the next slide. I used to have the Johns Hopkins list, but it's not accessible anymore. This is a fairly good guide to all of them. If you want to take a look at it, okay, I've tried to narrow down a few because there's something like 183 or 184 of them. Okay. But certain ones are going to be associated with certain types of cells. So a lymphoblast is going to have certain CD markers. A myeloblast is going to have certain CD markers. And a monoblast is going to have certain CD markers. Some will be common, but some will not. So if I have a population of blasts and I run it with my tagged antibodies, suddenly I can tell what types they are. Dr. Petterchek is one of our pathologists. First time I ever met the man. First day he's there, I take in a slide. I said, I got big ugly purple things because that's about as best as I could come up with on them. He took it, he says, oh, let's take a look at it. So he takes a look at it, he says, yep, those are ugly. I said, well, you're the pathologist and I need you to tell me what to write on the report that I'm going to give to the doctor because I can't just say big ugly purple things, which we would like to say a lot, but we can't. So he says, okay, well that'd be good. I said, no, I need you to tell me what to put in the report. Okay, so he writes out large mononuclear cells with lymphoid and monocytic characteristics, recommend flow cell cytometry to differentiate. You just said we got big ugly purple things and you don't know what they are. And he got paid $600 to say it. Okay, but basically, I don't know what they are. He says, I have no idea what they are. They're big ugly purple things. So we sent them for flow cell cytometry and they turned out to be something entirely not what we thought they were. They were lymphoma cells, which aren't even blasts. They were just ugly. This is much more characteristic than just identifying it by looking at it. Okay. Progenitor cells. CD34, highlight that one, circle that one, whatever you want to do. That's when you really want to make sure you know. It's going to come up again. When we get done talking about the different leukemias, I actually have a flow chart now that I have gotten from one of our affiliates, one of our clinical sites, and we'll go over it, but I want to do it after we do the leukemia so you know what I'm talking about. Yes?
So you're not going to see people use CD markers in the regular lab?
No, this is pretty much going to be more of a research lab, except, okay, here we go. CD4 and CD8. You're going to see them a lot. And actually several, the Sysmex does it, the Celldyne does it. Don't know if Coulter's do it or not. But this is one that actually you might see in a regular lab because that's the one we use to monitor AIDS patients. The CD4-CD8 ratio is what we use to monitor to see if HIV positive people have started to develop AIDS to determine whether they're going into the disease state or whether they're just carrying the virus. So that's what we watch to monitor our progress on those. Again, we'll get into a lot more of this as we go on. Yes?
It's possible to be an HIV carrier but not actually have the disease. So.
You have HIV, but you don't have AIDS. AIDS is the syndrome where you're active. Yeah, you can be seropositive for years and years and not actually develop the disease if you're given the right cocktail of antiviral drugs. All right, we like printing pictures. Okay. So basically, this is what one of the analyzers does. I think this one's the Abbott. I think this one is the Abbott flow cell cytometer. So if you look at this, so I may be measuring 5 or 6 different ones at a time. Each color represents a different CD marker that I'm looking for. Okay? We can read it at multiple wavelengths. We can use fluorescence. So I can measure multiple markers at one time to tell you. And these cells aren't going to have one marker. They're going to have a bagazillion. This is a little more close-up of what one of those looks like. This is actually just a flow cell cytometry from a basic blood smear. This is peripheral blood. These are your immature granulocytes. These are B cells and T cells. Okay, so it tells us not only what cell populations we have, which even our little XP will give you a basic differential. Yes.
Sorry.
No, don't be sorry. Please ask
You were
questions.
doing a diff. Couldn't you just stain all the different types of cells in different colors and then
It doesn't work that way. Your eyes cannot tell the difference between these colors. They have to be read by a spectrophotometer or a fluorometer. Yeah. And even if you were to stain them with all those different ones, you couldn't. Your eyes would not be able to differentiate between, you know, this shade of green and that shade of green. So there's gradients on it. And yeah, the biggest thing is money. Yes, could I put every single cell through a flow cell cytometer? Oh heck yeah. At
A little bit you could.
$700 or $800 a pop.
Yeah.
When you're in the lab, you guys aren't going to be looking at the normals. We'll talk about this at the end of the semester. But the automated differentials are getting better and better and better. We probably look at 1 out of 25 slides, tops, maybe 1 out of 50. And that's just the ones we just kind of glance at. Actual differentials, I don't think we probably do 10 in a week. Yes.
So this would be done off of whole blood, HTA whole blood. And why are they requiring also lithium heparin plasma?
It depends on your flow cell cytometer, exactly how it does it. There are different methodologies, and I'm trying to keep this like simple. It gets very, very, very complicated. And actually they don't, I don't know, they don't ask for plasma for flow cell. They might ask for heparin whole blood.
Oh, well,
Certain analyzers use heparin whole blood. But that just depends on your analyzer which one they want. And a lot of times what we're talking about, or what the application for hematology that I'm most concerned with here, is we're doing it on bone marrow. Okay? We're doing it on bone marrow. Cytogenetics, this is a different thing. This is not the same thing as flow cytometry. We're actually looking at the chromosomes. What you do, blast and progenitor cells are mitotic. Okay, that means I can make them make babies. They can still separate, they can still form new cells. Your segs, no. Once it gets to the segmented neutrophil stage, it is not able to reproduce. Okay? It's too old. All dried up, ain't gonna get, may have no more babies. Okay? Lymphocytes can be induced to clone. We already talked about that. So what I can do is I can make these cells reproduce. And then what I can do is I can add something to them. I add a mitogen and I say, grow, make more, be fruitful, multiply. Okay? And then when they get to a certain stage, I can toss a little something in there that says stop. And I can stop those cells in the midst of their reproductive cycle so that I can look at the chromosomes. Hopefully y'all know this. RNA and DNA, deoxyribonucleic acid and ribonucleic acid, they all pair up. The important thing here is that adenine and guanine always pair up, and thymine and cytosine always pair up. Okay. So we have AT and CG regions within our chromosomes. The nice thing is those stain a little bit differently with the stain that we use. That's nice. What do I expect you to know about this? Nothing. Okay. You need to understand that when it all reproduces, you make an extra copy, they line up. This and this is where we want to see. Okay, where everything's still lined up. Other than that, don't spend any time on that slide at all. Okay? What do we need to know here? What we actually do. This is an actual picture of chromosomes. We take the chromosomes, we kind of burst open the cell, we pull out the chromosomes, we line them up, and we stain them. The Q banding stains the areas that are AT rich, okay, or adenine thymine rich areas. The cytosine guanine areas, we then do a G banding stain, and we look for the AT areas will stain dark and the GC areas will stain light. C banding stains the centromere. That's nice. Basically, you need to remember that in Q banding, the AT areas stain And in G banding, AT is dark and GC is light. Can I look at that and tell you Jack? Nope. I can't tell you anything except that that's a boy. That's all I can tell you about this person is it's a boy. It takes tons and tons, as much time as we're gonna spend doing diffs, people have to spend learning this. Is this a good use of our time? No. Okay? But what you need to understand is that we can actually physically look at these chromosomes. What is really up and coming in the laboratory today, and this has been within the last 3 to 5 years at most, and that is now, just like we can take our antibody and hook something onto it, we can take a strand of DNA or a strand of RNA and hook onto it. And then we can go through and find genetic markers. We can actually go through, okay, last night I did what, seven Neisseria, gonorrhea, and chlamydia screens, where we actually take a labeled piece of DNA, put it into a sample. If there is chlamydia or gonorrhea present, because the sequencing matches up, it will hook onto that and it will tell me that those are present in the sample without me ever having to grow anything. Group A strep, I can do the same thing with that. Lots and lots. It's called molecular diagnostics, and it's becoming massive. And it is, right now, all we can do is look at the morphology, just like we look at diffs. But as this technology improves, just like adding the serology for the CD markers, we're going to be able to tell what's on the chromosomes a lot more specifically. And again, why don't we do that for everything? I can set up a Theramartin plate to grow gonorrhea for about, I think they're about $8-9 a piece. The molecular diagnostic test for it is, I think, $140. And that's just our cost to run it. That's not what we charge for it. That's just the cost to run it. So, but the price will come down. And it is getting closer and closer that you'll just be able to take somebody's blood, put it into a little machine, and it'll run all the chromosomes and genetics and tell you what's wrong with you. I don't know if it's good or bad thing, don't care. In situ hybridization, this is where we take one of those sequences. Okay, this was the earliest forms of being able to do molecular diagnostics. Okay, Barb is going to go over okay, if this is my strand of DNA, and we would use something that was 40 or 50 base pairs long, we're not going to use something that's 4, but I don't feel like writing that much today. What would be my corresponding pair over here?
TAGA.
Right? It's going to pair up in a known way. So I can create a probe. Then if I hook something on this so that I can see it, we create a complementary strand and we tag it. The first time somebody called down and asked for a fish test, I thought they were pulling my leg. What the heck is a fish test? What? You want a what? Diane, what's a, I don't know. So after we looked it up some, this is a method. So asking for a fish test is like calling down and saying, I need a blood test. Okay, you need to be a little more specific, please. Which exact test are we looking for here? This is called fluorescent in situ hybridization, or it is literally called a fish test. Okay? It's one of our older methods of looking for chromosomal abnormalities. Okay? We can detect specific gene sequences. This is nice, lovely. Direct DNA RNA detection, which is PCR. Again, I'm not going to ask you questions on this on the test at this point. Barb will be asking those questions when she gets you, and I will be asking more questions once you guys get to serology, because I have some massive research to do this summer. because there's a lot more methods now than just PCR. So I've got to do some research into those. That's nice. If you go online, they've got little animations, and they've got a little guy who goes along and hooks them all up. There's some really cute little cartoons on YouTube. Lovely. If it makes you happy, go for it. I'm not going to ask for it right now. Abnormalities. What do you need to know about these? Just the definitions. Okay. You need to just be able to define these. A multiploidy means I got more than one copy. I'm only supposed to have one copy of, but when I get done splitting my cell, I should only have one copy of each. A translocation, somebody was putting their Legos together and put one upside down. Okay, they got them backwards. I'm sorry, I put a piece in the wrong place. Inversion is where you put them together upside down. A deletion, I put something together and I forgot to put a piece in there. Okay, there's some parts left over. A duplication, the DNA copies, and it puts both copies into the same gene instead of splitting them in between two genes. Just basically, and they're fairly, multiploidy to many. I mean, they're pretty self-explanatory actually.
Is it multiploidy and duplication the same thing?
No, multiploidy means I have too many copies of a full gene. Duplication means I have a portion. I have multiple copies of a certain portion of the gene, not the full gene. That's just so that we can talk intelligently about separating out the leukemias. This is just to help you when we're starting to talk about all of these disease states that we're going to be talking about. This is actually, how many slides is this one? 19. Let's do at least a few of them. We may stop midway through. Malignant leukocyte disorders. So far we talked about qualitative and quantitative disorders. Just a few of the simple ones. When we are classifying leukemias, the first thing we have to do is split them out. Does anybody remember what our progenitor cell is, what our two main progenitor cells are for hematology? Progenitor cells, the step before those. I'll give you a hint. The lymphocytes come from CFUL. What does the other one come from? GEM. GEMM. CFU gem. I can guarantee you that's going to be on the test. Everything else comes from the CFU gem. We treat lymphoid leukemias with one type of treatment, and we treat myeloid leukemias with a different type of treatment. So separating them out based on what cell line they come from is very important, and especially in some of the new research stuff that's out there where they're able to target these leukemias even more exactly than they do right now. Once again, we just talked about all of this stuff. Identification needs a bone marrow aspiration and biopsy. days I'm going to grow 6 inches so you guys can reach the top. All right. To do a bone marrow. Any volunteers? This is going to be done by the pathologist, not by you. It's going to be done under sterile conditions. We have our lovely little setup. Somebody opened one, they didn't use it, so I kept it. It's probably about as sterile as your bathroom. What we do, the primary place we're going to go for bone marrow is right here. If you feel, even no matter how heavy you are, if you feel, you can find the tops of your hip. Okay. Most of our bone marrow production is done in the flat bones. Okay. So that's a nice, big, heavy, flat bone that we can get into. So what they do is they numb the skin. The nicer your doctor, the more numbing medication you get. Then they take and they numb the surface of the bone. Again, the nicer the doctor, the more numbing medication you get. Okay. Then we take this, which is known as a Jamshidi needle. Okay. You take it, well, the doctor takes it, makes a little cut in the skin, takes it down to the bone, and pushes it through until it pops, and he gets into the marrow space. Pull this out, put on a syringe. This is what hurts, because this is when the patients scream. You pull back, okay? And the thing about bone marrow is it's thick and it's got chunks in it. So you've all drawn blood with a syringe, right? Imagine trying to pull out something that's thick with chunks in it. So you pull back and this gives us our aspirate. Okay. We take this, we make slides out of it. Then the doctor, puts this back in. Yeah. And he continues to go down just a little bit. This is like drilling for oil. You know, when you drill, when they bring the drill out, there's a core within the drill. Do the same thing with this. Once you've got it firmly in there, pull this back out, twist it down in, wiggle, pull back out, then you take, let's see, I need anything, and push the core out. They then take the core to histology and slice it up to look at it. I never want to have this done, but God forbid if I have to. I want Dr. Pettercheck to do it. I don't want him to see my butt, but, you know. That man is so damn fast. He's awesome. Like, boom, boom. Okay, let's go to lunch. Yeah! And if you do 2 in a day, he buys you chocolate. But what this does is it gets the marrow out so that we can examine it directly. Okay. Honestly, from what I've been told, by non-wussy patients, this was his favorite. He's got a he's got a switchblade scalpel. Yes.
The core, what is the difference between the core and the marrow? What is the marrow is the core?
The marrow is the liquid portion that's kind of floating around in there. The core is actually the solid pieces of bone and stuff. And so you can see how it is arranged in there.
So the chunks that's coming out with the marrow is quite possibly part of the core?
Uh-huh. Yeah. I haven't got pictures of it. I'm working on it. That damn HIPAA stuff sort of, you know, it's like, it's not like I'm going to put patient names on it or anything. You know, it's like when I take stuff to the children's fairs, I've got a slice of brain, for God's sake, that we take with us. Now, it's from the 1980s, I think. Is it real? Yes. Is he dead? Yes.
He is now.
I know. That's not real brain. Yes, it's real brain. No, it's not. But that's how we do a bone marrow, okay? We're going to literally pull the marrow out. We're going to make slides. I have some slides you guys can look at. But that's how we perform one. And if you're lucky when you go to your clinical sites, you'll get to go see one. We do have students pass out on a nice regular basis. The first one I ever saw, the doctor I don't think was 4 foot tall. Looked like Yoda, I swear. He was having, great big woman, he was having trouble getting it. He's literally up on the stretcher on his knees. I lost it, had to leave. Yeah, it was bad. It was bad. Petrichek's really nice when he's, when you have someone who's, more full figured. He won't say I need because they make a longer jam sheety, he'll go. So leukemias are a group of diseases. Leukemia is not a disease by itself, it is a group of diseases caused by a malignant, unregulated proliferation of cells of the bone marrow. They involve blood-forming cells, but can and do often metastasize to other organs. Now, what does metastasize mean? Spread. What'd you say?
Paralyzed is what I'm thinking.
Yeah. No, metastasize. They go elsewhere. Okay, so it may start as a leukemia or especially lymphomas, and it may wind up in your brain, your liver, your lung, a lot of different places. Okay. The unregulated cells will replace the normal bone marrow. This is why we have to do a bone marrow biopsy. It can interfere with normal bone marrow function. It'll use up all the resources. It can invade other organs, and it will eventually cause death if not treated. Some of these will cause death quickly, and some of these can go a long time. Yes?
So, when somebody's getting a bone marrow replacement, is that going to cure it because everything's died out, or?
I'm going to ask you to be patient with me. We are going to talk about that in a couple of lectures, okay? Common symptoms. Basically, the symptoms are related to what's going on. Otherwise, infection, well, if your white cells aren't normal and aren't working, you're going to be prone to infections. Hemorrhage, you're going to bleed. One of the biggest ones that really we see a lot is patients come in with unexplained bruising. Okay, because as your bone marrow stops working, your platelets disappear first because they have the shortest life. Then your white cells disappear, and then it may take quite a while before your red cells actually start having a problem. Bone pain, well, ow, okay? If it's growing in there, and it's growing more than it should, it's filling the space and then pushing out, that's gonna cause pain. Splenomegaly simply means an enlargement of the spleen, because the spleen's job is to pull out all the old yucky cells. So if it's pulling out a lot of yucky cells, it's going to become enlarged. Clinical classification. What do we need to know here? Acute leukemia. The big keys here, circle, highlight, know that excess blasts greater than 20% by the WHO classification. This is the newer classification system. More than 20% blast in either the bone marrow or the peripheral blood is defined as acute leukemia. Okay. And the way I remember that it's acute is that kids are cute and blasts are very young cells. I told you I get stupider as we go along. Okay. We have usually a much more abrupt onset of clinical symptoms. Acute leukemia doesn't gradually sneak up on you. Pretty much you don't have it and then boom, you do. Okay? It is a rapid proliferation of very young cells, including an excess of blasts. It can occur both in children and in adults, although children tend to have acute leukemia. Children have acute leukemias, rarely do they have chronic. Chronic leukemias are generally in adults. Chronic leukemias have a much slower onset of signs and symptoms, especially when we talk about CLL or chronic lymphocytic leukemias. Those can take years. One of my friends had a husband who had CLL, And she says, looking back at his blood work prior to when he was diagnosed, he probably had already had it for 5 or 10 years before he was diagnosed with it, because it's a very, very slow, insidious kind of disease. These chronic leukemias almost always occur in adults. Like I said, I'll never say never, because somebody will find a case and it'll prove me wrong. But by and large, you're not going to find chronic leukemias in children. You will find acute leukemia in adults, though. Death can occur years after diagnosis, some of them 5, some 20. It just kind of depends on a lot of different things. We're generally going to see an elevated white count. I've seen CLL patients with white counts as high as 600 or 700. That's not normal. We're going to see an increase of mature white cells in the bone marrows, not blasts. Why? Because if we have more than 20% blasts, it's acute. We may see normal or increased platelet counts or decreased platelet counts. I don't know why that's in there. We will often see a little bit of anemia just as the white cells take over and there's not as much space for the red cells to be made. We will often see severe organomegaly, which means your spleen will enlarge, I'll be right with you, your liver will enlarge, and in CML especially, chronic myelogenous leukemia, you'll actually see it infiltrate the gums, so that their gums and their mouth will swell up. Yes?
When leukemia became really large after World War II, right? Is it because of the radiation that was going off that made the bone marrow mutate, or is it something else? Is that something else we're going to talk about later?
That's something else we're going to talk about later, okay?
That's something else we're going to talk about later.
And we're barely going to talk about it because honestly, there's still a lot of debate. How do we classify it? Pretty much just like I talked about any other case study. We're going to look at a history and a physical exam. We're going to look at the right stain smear, although especially with some of these chronic lymphocytic ones, your peripheral blood may be normal. You may have this massive lymphoma and your peripheral blood is absolutely normal because it's a solid tumor. We're going to do a bone marrow aspiration and biopsy. We're going to look at that. We're going to immunophenotype by flow cell, and we're going to do cytogenetics and genetic probes. Okay, that's pretty much standard for what we're going to use to diagnose leukemias. What slide are we on here?
About halfway.
We're about halfway. Why don't we just stop there? All right, it's about 9 o'clock. Let's stop there. We'll finish this up tomorrow. I don't really want to get into, we'll finish this tomorrow, and I think that's where we're going to stop for this week. Okay? Next week, because these next couple lectures, especially the chronic leukemias, they're deceptive. They're this many slides and this much information. So I'm going to try and split it into 2 tests so that we'll have just this kind of more basic stuff on this next test, plus some of the stuff from last week we're going to have on this test, and we'll talk more about that tomorrow. But I want to try and not, I don't want to get into the disease states yet. I think it's too much. I think it's going to be asking too much for you to try and start to remember. So next week, when you come in, you'll take the test, and then you really, really need to have your big girl and boy panties on, because we're really going to get into it. It is going to be a lot of material. A lot. There's tons of stuff, and I've broken it down as much as I can. I will try to, be as specific as I can on what you need to know. But what do you need to know? Basically, all of it. It's a lot of information. But like I said, I don't know if I'm a chart person. I learned really well from charts, so I did as many charts as I could. Be very careful going to YouTube on this. It's not something I ever had to tell students before, but there are some there's some stuff out there that is just flat wrong. And some of it is people with agendas and, so be very careful of your source when you're going to the internet. I had one student last year who Googled everything. Please don't, because there's some sources out there that are fantastic, and there are some that really, really are bad and just flat out wrong. And she argued with me, well, I Googled it. Well, what's your source? I don't know. I found it on the internet. Is that ever a good source? I found it on the internet. Well, there's people on the internet who tell you if you drink lemon juice every day. I swear I want to start something on the internet that says you should put dog feces on your feet every day, and that will clear up your complexion. It will detox your body and everything. And see how many people walk around with dog poop on their feet.
Be a new diet.
You can also buy tapeworms on the internet.
Are you serious?
What's really funny is snake oil salesmen in the 1800s marketed them as the miracle diet pill. And they do work. I mean, if you get tapeworms,